Hello, welcome back to the fourth video in the sequence going through the EDEXCEL Combined Science Biology Paper 1 Foundation Tier. So we're going to crack on and look at question 4. Okay, so question 4 is here. So on question 4 it says it says a student cut a piece of onion and placed it on the microscope slide. The student then placed his slide on the stage of a light microscope and looked through the eyepiece. No cells could be seen in the piece of onion, and the question is saying explain 2 ways in which this method could improve. So what I need is a statement and then a reason linked to that statement, and then a statement and then a reason linked to that statement. So let's just think. Student then placed it on the microscope and looked through the eyepiece. So the student actually cut the piece of onion themselves. So the first thing I can think of is they need to, what do they need to do? They need to have a single layer of cells, so they cut the onion too thick. Now the explanation linked to that is if the onion's too thick, then no light can pass through. And then the next part of the question, 2 ways. So the first way we've got is a single layer of cells, so they cut the onion too thick. Then no light can pass through, so that's the explanation. They've cut it, they haven't talked about staining it, so they could use a stain, and the reason why they could use a stain is then they could see the parts of the cell clearly. Other reasons you could have is I don't know if we're coming up with some other reasons. We could have the power of the lens is too low, so they won't be able to see the structures. So what they'll need to do for that is they will increase the magnification. They are the 3 most common answers that we generally find on this paper. Okay, moving on to the next question. Straight away I see, looking at this, it says it shows mitosis. So first of all, when I see mitosis, I always think of the phases and how to remember the phases. So I peed myself at the cinema. So we've got interphase, prophase, metaphase, anaphase, telophase and cytokinesis. Okay, so the 4 main stages. And then looking at the question here, we've got this is pointing at cell Q, and we've got something pointing at cell R here, and you can see something happening. You can see something like lining up in the middle of the cell in R, and then in Q you can actually see the chromosomes have separated and been pulled apart by the spindle fibers. Okay, so let's look at the question. The cells in figure 4 are taken from a rapidly growing part of the plant. Okay, so the rapidly growing part of the plant. The chloroplast is a subcellular structure, so an organelle, so it's definitely not the chloroplast, okay, because they're an organelle in the cell. The epithelium, okay, that's not that. The vacuole is another part of the plant cell, so it stores water, so it must be the meristem. Right, so the meristem is the area where you have rapidly growing or rapidly dividing cells. And then the next question says, which stage of mitosis is shown in cell R? So cell R is actually the one I highlighted in yellow. So in cell R, what I can see is that the chromosomes are lined up across the middle of the cell, waiting to be pulled apart. So it's cell R followed by cell Q if we're going through this. So let me just have a think about this. Going back to IP myself at the cinema. I stands for interphase. In interphase, what we have is the DNA replicated and the subcellular structures copied as well. In prophase, we have the spindle fibers start to appear and the nuclear membrane starts to break down, so it's not that. The next phase is metaphase. In metaphase, if I remember, the chromosomes line up, so it's definitely metaphase because the chromosomes line up across the middle. Then the next one, Q, is anaphase, because what's happening is the chromosomes are being pulled apart to each side, so it's anaphase. And if I remember rightly, we've got telophase. In telophase, what happens is the cell membrane, the nuclear membranes start to form around the chromosomes, and then cytokinesis, the thing finally splits up. Okay, so I IP myself at the cinemas, how I remember it. If I was doing this as an exam, what I'll probably do is under each little diagram is I'd draw a little picture to just explain it, and then I'd use that as my reference to answer the question. So the answer is metaphase. Okay, the next question, let's have a look at this now. It says, describe two genetic similarities of the new cells that will be produced by cell Q in figure 4. So cell Q in figure 4. Right, so it's about mitosis, because it's mitosis that happens in cell Q. So in mitosis, I know that we'll have the same or identical chromosomes, and then in mitosis the cells are diploid. Okay. One of the key questions is sometimes they'll ask you about mitosis, sometimes they'll ask you about meiosis, and understanding the differences between those two, but this question is just about mitosis. The cells in figure 4, so we're talking about figure 4 again. So these cells over this side of the page were heated in hydrochloric acid. So they're heated in hydrochloric acid. So the question's saying, state 2 safety precautions that should be taken when heating hydrochloric acid. So it doesn't matter what the context is. The question is a safety precaution that should be treated when heating hydrochloric acid. So safety precautions, first thing is wear goggles. So wear goggles. And the second thing is you could use water bath to heat the hydrochloric acid, the hydrochloric acid, the HCl. Alternative answers as well is you could wear gloves. I suppose that would be an adequate safety precaution as well. And the final part of this question, okay, it says explain one advantage of using an electron microscope to observe plants. Now the key thing here is, because it says explain, I need to give a statement and then explain that statement. Okay, so an electron microscope. So my statement is going to be electron microscopes have a, let's get a pen to write with, electron microscopes have a higher resolution, and my explanation of that is so more detail can be seen. Okay, and that draws us to the end of our video for this question.